Cell junction.
Let's learn what is cell junction. Cell junctions are contact points between the plasma membranes of tissue cells. Most epithelial cells and some muscle and nerve cells are tightly joined into functional units. There are 5 types of cell junction: tight junctions, adherens junctions, desmosomes, hemidesmosomes, gap junctions. Tight junctions. Tight junctions are also called occluding junctions and zonale occludants due to their structure and function. Occluding junctions, this name emphasizes their primary function, creating a seal between adjacent epithelial cells to occlude, block, the passage of molecules and ions between cells. Zonale occludants. This term comes from Latin. Zonula means small belt, or band, indicating that tight junctions form a continuous, belt-like structure around the cell. Occludants means occluding, or blocking, referring to their role in preventing leakage between cells. These are multiprotein junctional complexes which forms the web-like structure between epithelial cells, sealing and preventing leakage of solutes and water. They inhibit the passage of substances between cells and prevent the contents of these organs from leaking into the blood or surrounding tissues. Adherens junctions. Adherens junctions or adhesion junction or anchoring junctions contain plaque, a dense layer of proteins on the inside of the plasma membrane that attaches both to transmembrane proteins and to microfilaments of the cytoskeleton. Plaque contains of the catenins. Catenins are of 4 types. Beta catenins, P120 catenins, alpha catenins, gamma catenin that is, placaglobe. Beta catenin links cadherins to alpha catenin, which connects to the actin cytoskeleton. P120 catenins stabilizes cadherins at the membrane by preventing the degradation. Alpha catenin interacts with beta catenin and directly binds actin filament. Gamma catenin that is, placoglobin. Similar in structure to beta catenin but functions in both adherens junctions and desmosomes. Transmembrane glycoproteins called cadherins. Cadherins are a family of calcium-dependent transmembrane proteins. Join the cell. Structure of cadherins consists of extracellular domain, transmembrane domain, cytoplasmic domain. Extracellular domain contains multiple cadherin repeats that bind to cadherins on adjacent cells in a homophilic manner, that is, E cadherin binds to E cadherin, N cadherin to N cadherin, etc. Transmembrane domain anchors the cadherin to the plasma membrane. Cytoplasmic domain interacts with catenins, beta catenin, P120 catenin, to link cadherins to the actin cytoskeleton, forming adherens junctions. Each cadherin inserts into the plaque from the opposite side of the plasma membrane, partially crosses the intercellular space that is, the space between the cells, and connects to cadherins of an adjacent cell. In epithelial cells, adherens junctions often form extensive zones called adhesion belts because they encircle the cell. Similar to the way a belt encircles your waist. Adherens junctions help epithelial surfaces resist separation during various contractile activities, as when food moves through the intestines. Desmosomes. Like adherens junctions, desmosomes contain plaque and have transmembrane glycoproteins. cadherins, that extend into the intercellular space between adjacent cell membranes and attach cells to one another. However, unlike adherens junctions, the plaque of desmosomes does not attach to microfilaments. Instead, a desmosome plaque attaches to elements of the cytoskeleton known as intermediate filaments, which consist of the protein keratin. The intermediate filaments extend from desmosomes on one side of the cell across the cytosol to desmosomes on the opposite side of the cell. This structural arrangement contributes to the stability of the cells and tissue. These spot weld-like junctions are common among the cells that make up the epidermis, which is the outermost layer of the skin, and amongst cardiac muscle cells in the heart. Desmosomes prevent epidermal cells from separating under tension and cardiac muscle cells from pulling apart during contraction. Hemidesmosome. Hemidesmosomes are like tiny anchors that attach skin and other epithelial cells to the underlying basement membrane. They prevent cells from peeling off under mechanical stress, like rubbing or stretching. Hemidesmosomes, hemi means half, resemble desmosomes, but they do not link adjacent cells. The name arises from the fact that they look like half of a desmosome. However, the transmembrane glycoproteins in hemidesmosomes are integrins, rather than cadherins. Structure of hemidesmosome. Transmembrane proteins, the hooks. These proteins extends from one side of the cell membrane to the other side the cell membrane and grab onto the basement membrane. Main protein. Integrin works like a hook to latch onto laminin 332 in the basement membrane. Another protein, collagen 17, helps reinforce this connection. Plaque proteins, the connectors. Inside the cell, special proteins form a plaque, a dense structure, that connects the hooks, integrins, to the cytoskeleton. Main proteins, plectin and BP230. These act like cables, linking the integrins to intermediate filaments, keratin, inside the cell. This connection makes sure the cell stays firmly attached and resists stress. Basement membrane, the landing pad. Below the cell, the basement membrane is like a landing pad where the cell anchors. Key protein, laminin 332. This interacts with integrins to form a strong attachment. Collagen 4 provides extra support to stabilize the attachment. Why hemidesmosomes are important. They prevent skin and epithelial cells from peeling off under friction. They keep tissues stable and strong, especially in areas like the skin, cornea, and esophagus. Gap junction. Gap junctions are specialized cell-to-cell connections that allow direct communication between adjacent cells. They enable the exchange of ions, small molecules, and electrical signals, making them essential for coordinated cellular activity, especially in the heart, nervous system, and smooth muscle. Structure of gap junctions includes connexins, gap junction pore. Connexins. These are transmembrane proteins that come together to form a connexins, hemichannel. There are different types of connexins. Examples: connexin 43 in the heart. Connexins. Each connexin is made of 6 connexin proteins. A connexin from one cell aligns with a connexin from another cell, forming a continuous channel. Gap junction pores. The plasma membranes of gap junctions are not fused together as in tight junctions but are separated by a very narrow intercellular gap. These pores allow the passage of small molecules like ions, nutrients, second messengers, but block larger molecules like proteins and DNA. Functions of gap junctions. The transfer of nutrients, and perhaps wastes, takes place through gap junctions in avascular tissues such as the lens and cornea of the eye. Gap junctions allow the cells in a tissue to communicate with one another. In a developing embryo, some of the chemical and electrical signals that regulate growth and cell differentiation travel via gap junctions. Gap junctions also enable nerve or muscle impulses to spread rapidly among cells, a process that is crucial for the normal operation of some parts of the nervous system and for the contraction of muscle in the heart, gastrointestinal tract, and uterus. Like, share and subscribe for more updates.